Hello. Thank you everyone for coming. It's my great pleasure to introduce our seminar speaker today in the genetics program. David Greenstein is a professor in the Department of Genetics, Cell Biology, and Development at the University of Minnesota. So I tried to prepare a brief introduction, but we'll see how successful I was. So he got his bachelor's from the University of Pennsylvania in chemistry and biochemistry. Then he moved on to the Rockefeller University to get his PhD on phage replication, working in the lab of Norton Zinder, which has a connection to UW because he was a student in Joshua Lederberg's lab while he was here at UW. And actually, when David did his PhD at the Rockefeller, Joshua Lederberg was the president of Rockefeller at the time, too. So there's a UW genetics connection. Um, and then he saw the light and moved on to do his postdoc at Harvard with Gary Ruvkun before he was a Nobel laureate. And when he was actually like in the early days of his lab, David was one of his first postdocs. And in Gary Ruvkun's lab at Harvard, he developed his research program studying oocyte biology and early embryogenesis. And he's still going strong on this topic. So he got his first faculty position at Vanderbilt University as an assistant professor. By the time I joined Vanderbilt as an assistant professor, he was an associate professor. And for the whole time since I joined Vanderbilt to now, he's been a great colleague. He's been the main mentor throughout my life. I don't think I would be where I am now if it wasn't for all the good advice he gave me about like lots of things in the world of science. So he has a very special place in my heart and in my academic career. Um, and actually, when I started there, like I asked to join their lab meetings because I was so lonely as an assistant professor with nobody to talk to. So I was a guest at their lab meeting for a while, my lab and I. And David has a really strong history also, as I just mentioned, he has been a great mentor to me, but he has an all around a very strong history of mentoring, teaching, and service. Very involved in the Genetic Society of America in various roles. He's also had major administrative roles at his university. So he's really contributing all around to the field of science. But above all, David is a really creative and rigorous scientist, and his lab has been making major contributions to the germline research field. So, for example, he discovered like early on in his independent career that some cytoskeletal proteins involved in motility of the amoeboid sperm in C. elegans were actually being released from the sperm to induce oocyte meiotic maturation and ovulation. And he spent many years and still studying the downstream mechanisms from that. And he's also looked at the role of gap junctions in mediating soma, germline interactions, translational control during meiotic maturation and oocyte to embryo transition, etc. And more recently, he has expanded his research to leverage the C. elegans oocyte to study human diseases. And I think today we'll hear a little bit about different projects in the lab, but I'm really excited to hear his talk, and it's really a pleasure to host him here. Thank you.
Thanks, Daniela. That was really, really lovely. Thank you. Um, I've had a wonderful day, meeting colleagues and students and hearing about the exciting work that's been going on here, and I'm excited to tell you about what we've been doing. Please feel free to ask questions during the seminar. And I'm going to try to go with the lights like this, but if you need them turned down, let me know, because hard for me to see from where I'm sitting or standing. Okay, so these remarkable drawings were made by Hermann Fall over 150 years ago. These are drawings of fertilization in the starfish, and he describes this process in detail. He was among the first people to observe fertilization. Sadly, his career was cut short, not due to lack of funding, but rather he was lost at sea in his research vessel, the Astor. But I like to think that, if he should miraculously come back today through the Bermuda Triangle, he would be really amazed at how the field has progressed to getting molecular level descriptions of gametogenesis, gametic interactions, and early post-fertilization development. I think you'll be really pleased that even today, the starfish is really a premier model for its studying these events because of its contributions that are possible because of biochemistry and cell biology. But I like to study this beautiful animal, the elegans, which you have wonderful colleagues here who are doing amazing work. So I don't have to tell you about all the tools. But for me, the two things that really, set this, make me want to study this organism is we can do genetics. So we can do genetics, both forward genetics to discover new players in the pathway. And also it keeps us honest. The genetics underpins everything we do because the worm doesn't lie. And finally, as you can see, the worm is transparent. So we can look at the events of oocyte development, fertilization, and early embryogenesis in intact living animals. Seeing is believing, at least for me. So today I'm going to tell you two stories, and these are highly collaborative stories that were begun during the pandemic, and we reached out and we collaborated with Erin Osborne Nishimura at Colorado State University. I'm going to tell you a story about the translational regulation and the control of maternal mRNA clearance in the embryo, and a second story with Amy Jaramillo Lambert from the University of Delaware on a sperm oocyte protein partnership that's required for early post-fertilization development. Oops. So the oocytes of most sexually reproducing animals arrest in meiotic prophase, and they often arrest for many years, for example, up to 50 years in humans. And in response to hormonal signaling, they undergo this process of meiotic maturation. CDK gets activated, they undergo M phase, and then they often are ovulated and fertilized, and the meiotic divisions are completed afterwards. In C. elegans, the hormonal signal actually comes from the sperm, and it's the major sperm protein hormone. And the meiotic divisions happen after fertilization here. Now, the oocytes in most species are transcriptionally quiescent. So translational control is really important, and zygotic transcription starts in the embryo at different times, depending on the species. In this species, zygotic transcription starts at around the 2 or 4 cell stage. And what this means is the process of meiotic maturation has to set in place a process called maternal mRNA clearance, which is important for setting the transcriptomes to get rid of the mRNAs that were early acting to set the stage for zygotic transcription. So I'll tell you a little bit about this. But as I said, it's really, really wonderful is that we can observe these processes in intact living animals. And so what we're seeing here is the oocyte, and this is the nucleus of the oocyte, and the sperm is over here, and the sperm and the somatogonadal cells, which are contracting into the sheath cells, are receiving this signal, and this oocyte will undergo meiotic maturation, nuclear envelope breakdown. CDK gets activated, we ovulate and fertilize. And so I took this movie in about 1996, and when I was doing this, some of the things I wanted to know were, what's the signal? Why does this oocyte respond but not this one? What's the role of translational regulation? How do the events that happen here set the stage for what happens next? So those are some of the things that we've been doing over the years. So as Daniela mentioned, and as I just said, the sperm release a hormone that triggers meiotic maturation, and it's this molecule with a double life called major sperm protein. It's the most abundant protein in sperm, which was defined by Sam Ward, who made this movie of the sperm crawling. So nematode sperm are, highly derived. They move by crawling, and they use this MSP cytoskeleton. And what we showed was that a small fraction of this major sperm protein is released in extracellular vesicles, about 300 nanometer extracellular vesicles, which are unstable, and then release MSP in a soluble form where it can trigger oocyte meiotic maturation, sheath contraction, and the flows that drive oocyte growth. So downstream in the process, two RNA binding proteins are important for regulating the process of meiotic maturation, oocyte development, oocyte growth, and setting the stage for the oocyte to embryo transition. And these are Lin41 and the OMA proteins. So actually the three proteins, the two OMA proteins, they're paralogs. And so these proteins function antagonistically to one another. So Lin41 is a trim NHL RNA binding protein, and it functions to prevent meiotic exit from meiotic prophase and to promote oocyte growth, whereas the OMA proteins promote oocyte meiotic maturation and inhibit oocyte growth. Despite this antagonistic relationship, these RNA binding proteins co-purify in large RNPs, and they regulate batteries of genes that are important for oogenesis and the oocyte to embryo transition. So late in oogenesis, Lin41 is inactivated as a translational repressor and is degraded upon CDK activation during meiotic maturation. So I think you can see in this movie, we're looking at Lin41 GFP when this is sort of the dynamic version of the movie I showed you before. The protein is inactivated and rapidly degraded. So as I mentioned, these proteins co-purify in large RNPs, and these RNPs are pretty stable, and they can be isolated on glycerol gradients, and we can determine the RNAs that are associated with them. And so, for example, by RNA sequencing. So this group of RNAs is enriched, fourfold with P is, with a highly statistically significant level. And we find Lin29, which is, oops, this pointer just died. I can use this pointer, probably. Lin29, which is the famous transcription factor in the soma, but it's Lin41 has a role in the soma also in the heterochronic pathway. But this is not present at high levels in our IPs. But the gene I'm going to talk to you about, SPIN4, is both highly enriched and abundant. There are also RNAs that associate with both proteins, as well as RNAs that associate with the OMA protein. So we've identified three modes of translational regulation, and this one is one of my favorites. This is an example of genes that are repressed by Lin41, but the OMA proteins have no role in affecting their translation. And I love this example because this is the largest subunit of ORC, the origin recognition complex. And the reason I like this example is if you were setting up the system, this is how you might design it. So what you can see here is ORC is expressed in the region of where the stem cells are located, where there's mitotic S phase and premeiotic S phase. And then Lin41, that's over here, sorry. And Lin41 represses it. And then all of a sudden, after Lin41 is inactivated as a translational repressor, you get ORC, so you can have origin-dependent DNA replication in the embryo. There are also genes like this one that are repressed by both factors. The mode of control that we find most interesting and might relate to the antagonistic role of the Lin41 and the OMA proteins is a gene like SPIN4, in which Lin41 represses its translation and the OMA proteins promote its translation. SPIN4 encodes an RB-FOX RNA binding protein, and it's required for embryonic development. So late in development, when Lin41, oocyte development, when Lin41 is inactivated as a translational repressor, this leads to a reciprocal expression pattern where spin4 comes on in the maturing oocyte or slightly before and is abundant in the embryo. And the story I'm going to tell you is that we've discovered that spin4 now controls the process of maternal mRNA clearance in response to meiotic maturation, because we've relieved the repression by Lin4, and spin4 actually, one of its targets is actually Lin41. And this contrasts with the famous case in the soma, in which actually the Let7 microRNA in the soma represses Lin41. This doesn't appear to happen in the germline. And this is important both for C. elegans heterochronic development, but also in human cells in the process of reprogramming. So Tatsuya Tsukamoto over here in the lab wanted to know what spin4 was doing late during oogenesis when it was first expressed in the oocytes. So he purified spin4 containing RNPs to determine the RNAs that are enriched in these RNPs by RNA sequencing. But he made a really important choice, which at the time we didn't appreciate why it was so important, but now we do, is he decided to do this purification from sterile strains of worms in which no sperm was present, because he wanted to understand what spin4 is doing right when it's being made and not what's happening later in the embryo. And it turned out this was a really good choice, because as I'll show you, many of the RNAs it associates with are degraded in the embryos, and this would not have been very successful. So in all, he identified about 728 RNAs that associate with spin4, and many of these also associate with Lin41 and the OMA proteins. So a key insight came from Carolyn Spike, who's over here. She noticed that many spin4-associated mRNAs are downregulated and degraded in one-cell embryos relative to oocytes, as reported by the Rajewski lab in this paper. And this includes RNAs that are repressed in oocytes by spin4, proteins RNA for proteins that regulate spin4 expression in oocytes, and many early acting genes that are required for eggshell synthesis, which was one of the first things that happens after fertilization. And so we had this great collaboration, we still do, with Aaron Nishimura's lab and Dylan Parker over here, who's now in Roy Parker's lab, but no relation, wanted to look at the level of many of these targets by SM fish. And what I think you can see here is that the Lin4 mRNA is downregulated as oocytes, as embryos progress from the 2 to the 8 to the 16 cell stage. And this downregulation doesn't happen when spin4 is depleted. As a technical note, if you notice, we start our analysis here with 2-cell embryos, and that's because to get high throughput, we actually use embryos by alkaline hypochlorite treatment, and the 1-cell embryos don't survive. But what we think is happening is actually the mRNA is starting to be cleared for this gene, actually at the 1-cell stage or slightly before. So he looked at several targets. Here we're showing Lin41, chitin synthase, which we'll talk about a little later, CpG2 and OMA2. And in each case, these spin4-associated RNAs are cleared from developing embryos in a spin4-dependent fashion, whereas non-targets are not affected. What we also notice is that the timing of it seems to be target specific. So for example, for Lin41 or chitin synthase, it happens before the 2-cell stage, at least, as it starts to happen, whereas it occurs a little later for these two genes, CpG2 and OMA2. In all, 9 of 12 spin4 associated transcripts were inefficiently cleared in embryos. And this suggests to us that probably the highly pleiotropic effect of this mutant, in which cell divisions are messed up, eggshell formation seems to be messed up, it seems to be very early and pleiotropic, may just result from an imbalance in the transcriptome because of a failure to clear many mRNAs. So it turns out that RB-Fox translation, RB-Fox proteins have been very well studied, especially nuclear RB-Fox proteins that are involved in alternative splicing. So this protein, SPIN4, is cytoplasmic, but the field has found that these RB-Fox proteins bind a consensus site. And we noticed, actually Carolyn Spike noticed, that in the Lin4 3' UTR, Lin41 3' UTR, there was a single Fox site. So we can use genome editing to delete the site. And what Nolly Torres and Carissa Coleman found was that when we delete the site, there's a defect in clearance of the Lin41 mRNA, further linking SPIN4 to this process. And they did some quantifications, including an analysis of neighboring sequences here. And what we see is that the small Fox deletion has an effect, but neighboring sequences seem to contribute. And so there may be other things involved in recruitment of machinery to cause the degradation, and we're still trying to understand this. So Erica Tsukamoto in the lab, a very talented undergraduate in the lab, wanted to see whether SPIN4 can directly bind to targets, and she was very successful in purifying a portion of the SPIN4 protein that has the RRM, and she showed that it could bind small RNA pieces that have this binding site, and it could have a modest KD. So we think that for many of the targets of SPIN4 mRNA clearance, it may just be simply recognition that plays a key role, RNA recognition that plays a key role. Well, Erica, being a very talented student, I asked her whether she would take the project of determining the mechanism of SPIN4 dependent mRNA clearance. And we thought that if we asked the worm and asked very nicely, she would tell us how it worked. So the next slide will show sort of the logic of how she went about devising a genetic screen to get at the mechanism. So PUF3 and PUF11 are two nearly identical familial related RNA binding proteins that have been discovered and described by Judith Kimball's lab for their important role in stem cell maintenance. And here you can see they're expressed in that region, but they're also expressed in oocytes and quite abundantly. And what they also noticed was that PUF3, PUF11 double mutants are maternal effect lethal. What we noticed is that in these oocytes of PUF3, PUF11 mutants, SPIN4 comes on maybe an hour earlier than it should in oogenesis. And we wondered whether premature RNA clearance in the oocytes might cause this maternal effect lethality. Okay? So the funny thing about these mutants that was first reported by the Kimball lab was that even though they're maternal effect lethal, occasionally an embryo will hatch and will grow up to adulthood and look happy, only to have the same effect, to be maternal effect lethal. So it's kind of on the edge, and it seemed like it was kind of good for a genetic screen. So what Erica did is she sought dominant suppressors of PUF3, PUF11 maternal effect lethality. The idea is maybe she's going to get factors that are involved in maternal mRNA clearance. And she got rare suppressor mutations, but these suppressor mutations come out pretty much at knockout frequency. So we think here, this is dominance due to haploinsufficiency, to dosage of factors that may be in the pathway. Okay? She got about 19 mutations in about almost 30,000 haploid genome screen. And given that she was doing her undergraduate coursework at the time, it's just pretty amazing. So one of the mutants she got out of this was she got a stop codon in spin 4. So this was very reassuring. So this is consistent and validates sort of the idea of the screen. She also got alleles of the CCR4 knot complex. And so you got 8 alleles, and including 6 nonsense alleles in the largest subunit of the CCR4 knot complex. She also got missense alleles near the active site of one of the 2 deadenylases in the complex. So the CCR4 knot complex deadenylates mRNAs and can cause their turnover. It also participates in translational repression. So naturally, we wanted to determine whether the CCR4 knot complex is required for maternal mRNA clearance. The problem here is that these are essential genes. They're required in many cells during development, and they're lethal. So what Carolyn Spike did is she used the auxin-inducible depletion system to deplete components of this pathway so that she can grow the embryos to the worms to adulthood, get oocytes, do a 3-hour depletion essentially, and then look at transcript levels. So what we're showing here is we're looking at 2 targets. We're looking at Lin41 and chitin synthase. And these are spin4 associated mRNAs. And what we're seeing is 2-color SM fish for them, and also set3, which is a control. And I think you can see that after LET7-11 depletion, Lin41 mRNA levels are elevated, implicating the CCR4 knot complex. And so Carolyn, being the kind of scientist she is, she wanted to do this quantitatively. So she looked at wild type, and she sees, as I showed you, Lin41 levels decline as embryos progress from the 2 to the 4 to the 8-cell stage. I should say she also did this for chitin synthase. But when she deletes LET7-11, the mRNA levels stay high and embryonic lethality results. It turns out that when you just tag LET7-11 and when you tag LET7-11 and look, even in the absence of auxin or the TUR1 driver, it's a viable hypomorph. And in this instance, even though these worms, we see a decreased kinetics, we actually have made many CRISPR lines in the lab, hundreds of CRISPR lines. We have like a theorem that any tag you make is always different from the wild type. And there will be a genetic test that can reveal this difference. You may not know what that genetic test is, but we're finding this to be true. That what evolution gives us is always better than our tag version. So this is an example in which we can see that even though the process of maternal mRNA clearance is perturbed, it's not doesn't negatively impact viability so much. We got a similar thing. CCF1 is one of the 2 deadenylases in the complex. In her genetics, Erica isolated a hypomorph, which is viable and fertile, very good brood size. It deletes 2 amino acids near the active site, but we see delayed kinetics of Lin41 mRNA clearance. And we get a similar effect when we tag CCF1 with AID and just look at the tag. It also is viable, yet we see a delayed kinetics. When we actually deplete CCF1, we get a stronger effect, but it's not as strong as deleting the scaffold LET7-11. We think that's because there's 2 deadenylases in the complex, CCF1 and CCR4. And it turns out CCR4 null alleles are actually viable and fertile. They show effects on the kinetics of maternal mRNA depletion. And as you might suspect, when you combine CCR4 null allele with this CCF1 allele, you really can't grow the worm. So there's redundancy in the deadenylase complex, but probably for this process, CCF1 is probably the major player. Of course, when she looks at set 3 controls, none of the effects, none of the things that I showed you have any effect on the outcome. So I showed you evidence that the translational regulators of meiotic maturation set the stage for early embryogenesis by regulating the process of maternal mRNA clearance. And this fits well with studies in other systems. So for example, in Drosophila, SMOG recruits the controls the RNA binding protein SMOG, controls the process of maternal mRNA clearance of a subset of messages and recruits this complex. And in mouse, it's BTG4. And in zebrafish, it's actually zygotic microRNAs that play the important role. And so some of the questions we want to study in the future is what controls the recruitment of this complex and its activity to turn over a subset of messages. I mean, now I'm going to switch gears and talk to you about another collaborative project that also arises from the study of the RNA binding proteins that we are studying. And this is about a sperm oocyte protein partnership. And this is work with, it's going to be a long-standing collaboration with Amy Jaramillo Lambert. And so what Tatsuya noticed is when he made a Venn diagram of looking at RNAs that associate with SPIN4, Lin41, or OMA proteins, he had many famous genes in the C. elegans germline and early embryogenesis field. They have really cool names, not as cool as the fly names, but really names because people gave them names because they did something important. But what he noticed was that about 50% of the genes that were associated with mRNAs that aren't even shown here are uncharacterized, and they may too be important. So he looked in knockout databases, and he found 90 genes that were annotated as being lethal or sterile in knockout databases, and he focused in on one gene that we're calling OOPS1 for reasons that will become apparent. And I think Tatsuya deserves a lot of credit and for his courage for studying this gene, because it is an intrinsically disordered, codes an intrinsically disordered protein with no homologs outside of center-abdited nematodes. Okay, so the death knell for the NIH study section. So, but on the other hand, the reason he wanted to study it is because the phenotype told him it was doing something really important. And so what he found was OOPS1 null mutants do not complete meiosis. They fail to form polar bodies. They have a defect in eggshell formation. They exhibit polyspermy, so more than one sperm can enter. There are no embryonic divisions, and the defect is in the oocyte. And here you can see, here's wild type, and here's some embryos in the uterus, and you're starting to divide. And here you can see there's these like cells that are just sort of stacking up in the uterus, and we know they're fertilized. They don't make eggshells, and nothing happens to them. So we thought this was something to study. So Kent Kwa in Amy Jaramillo Lambert's lab made movies of what happens in the wild type and OOPS1 mutants. And so what we're looking at here is tubulin in green and histones, which light up the chromatin in red, and we're looking at the wild type. So meiosis, the oocyte meiotic divisions are happening after fertilization, and you get a barrel-shaped spindle, and you form the first and the second polar bodies. The second polar body will be out of the plane of focus in this movie. So here's the first division, and then there's the second division coming up. And of course, these are done in utero, so things kind of move around a little bit. And as I said, OOPS1 mutants fail. And so one of the phenotypes we see is what's called the completion phenotype. What happens is the chromosomes will segregate it at meiosis 1, but meiotic cytokinesis fails, and polar bodies don't form. So, now we're seeing the second division. So here, chromosomes are segregating, but then, oops, the chromosomes come back on the spindle, and then they segregate, but they remain interior, and this leads to the formation of multinucleate in a polyploid embryo. The second phenotype, so this is only 50% of them. The second phenotype we see is meiotic arrest. And this is it can happen at meiosis 1 or meiosis 2. And this is an example of an M2 arrest. And so here you can see the chromosomes segregate. They have a little trouble, but the chromosomes then pop back on the M2 spindle, so they didn't form polar bodies. And then the spindle becomes disorganized, and meiosis arrests. Okay. So the breakthrough for us came when Tatsuya did tandem affinity purification of OOPS1. And so we've done many of these now, and I think it's fair to say that he's developed into sort of an art form. And so he got a striking result. So when he brings down OOPS1, and we're looking at mass spec data, he got Sp11. And this was really remarkable because Sp11 is one of the few described paternal effect lethal mutations in the worm. It was discovered by Steve Lernoux in 1988, and Susan Strom did beautiful work on it back in Browning and Strom in 1996, but the mechanism of action wasn't known, and no one really studied it. And so this was kind of remarkable because normally when we bring down a protein for mass spec, that's the thing that's most highly covered. But you can see when we bring down OOPS1, we get really good representation of Sp11. So we think these proteins interact probably after fertilization in a diffusion limited mass in a very efficient way. We also got these two phosphatases. These are PP1 homologs. They basically are nearly identical to human PP1, and they're paralogs that are specifically expressed in the sperm. He did this the other way around with Sp11, although you can't compare these numbers to the first ones because we upgraded our mass spec machinery between these experiments. But you can bring down Sp11, you get OOPS1, and you get GSP3 and GSP4. So we thought that this is maybe a sperm protein partnership. And what's really kind of intriguing is to our ability to see these proteins in vivo. We think, we know genetically that the OOPS1 is required. OOPS1 stands for oocyte partner, Sp11, by the way. We know that OOPS1 is required in the oocyte. Sp11 is required in the sperm. And you can see by expression, they really don't overlap except after fertilization. So they're only found in the same cell after fertilization. So the other kind of strange thing is that because phosphatases were there, I was wondering if these were phosphoproteins. And so when we took a careful look at the mass spec, we found that both of these proteins are heavily phosphorylated. And we don't know, we think this may play an important regulatory role, but we don't really understand it. We did these purifications from whole worms. But when we express the proteins in bacteria and purify them, they co-purify as a complex, showing that they don't need any other C. elegans proteins to interact. So we think this is probably a high affinity interaction that forms only after fertilization. So remarkably, Sp11 null mutants phenocopy OOPS1 null mutants. So we get the same phenotypes. We get about 50% forming this completion phenotype where all the chromosomes segregate, but they don't form polar bodies and they remain inside the embryo. And about 50% of the embryos are resting at meiosis 1 and meiosis 2. And we see the same phenotype in the double mutant. So it's consistent with the idea they function in the pathway. Sort of the curious thing is Frank McNally had previously analyzed this allele Sp11HC90, which is a stop codon in the middle of the protein. And I think he recorded 5 animals, and he found that all 5 of them did this completion phenotype, and none of them arrested. And we repeated that looking at many more, and we found that's actually true. And to actually get a null mutant, we had to delete the whole coding sequence. So a little bit of this protein may go a long way, even though, so even a stop codon in the middle of G is not null here. So it really highlights the importance of getting null phenotypes and knowing you're looking at null phenotypes. So I told you that OOPS1 and SPE11s are defective in meiotic cytokinesis. And so this is a really interesting asymmetric cell division that forms a big cell, the embryo, and, and small polar bodies. And it's an actin-based process, and the cytokinetic ring forms at the anterior. And so I thought that, the field is coming to the idea that in addition to microtubules, cortical actin dynamics are really important for completing oocyte meiosis. So I had the idea that maybe this complex is an actin regulator. And my good friend and colleague, Naomi Quartermanche, is a biophysicist and biochemist who studied the actin cytoskeleton. And somehow I was able to convince her to do some experiments with me. And it turned out her idea was correct. So this shows actually a very simple experiment in which we polymerize actin together, either in the presence or absence of OOPS1 SPE11 complex, and isolate the actin by centrifugation. And ordinarily, OOPS1 SPE11, here looking at SPE11, goes to the supernatant. But when we do this experiment in the presence of actin, it now goes to the pellet. It's distributed between the supernatant and the pellet. This tells us that the complex can interact with actin in the absence of other proteins. The other thing we noticed in this experiment was that this is actin alone, this is in the supernatant, and this is the pellet. When we have OOPS1 and SPE11, we appear to get less of the less polymerized actin form. So we wanted to do quantitative bulk kinetic experiments to analyze this. And so this shows just a time course of polymerization, the presence of different amounts of the complex. And what you can see is even though these reactions haven't reached equilibrium, it looks like the rate of polymerization is lower. So what we did next is, usually these assays, the rate limiting step is nucleation of the actin filaments. So we did assays in which we used constitutively active formins, which accelerate the rate of polymerization by promoting nucleation. And we noticed that when we add different amounts of our complex, of SPE11 OOPS1 complex, we get a decreased rate of actin polymerization, yet the plateau level is the same at the end, which suggests to us that this complex affects actin nucleation. I should say we can rule out the idea that it's simply sequestering monomers, because these experiments are done at about 200 to 50-fold less amount of complex, SPE11 OOPS1 complex, than monomers. So what we hope to do next is to do single molecule assays to explore this activity further, as well as looking more at the dynamics in vivo of the actin cytoskeleton. So I mentioned that OOPS1 and SPE11 are intrinsically disordered proteins using different predictions and AlphaFold. But what Katie Wallstrom noticed is that if she used AlphaFold multimer and puts the proteins together, now there would be well-ordered regions. So for example, in the top model, you're seeing SPE11 in blue and OOPS1 in pink. And it seems that SPE11, according to this model, would wrap around OOPS1. And then here we're looking at using the AlphaFold colors, where blue is regions that are predicted to be better ordered. Now, we were very skeptical of this, and we think that this is no substitute necessarily for doing structural biology, but remarkably, it was supported by genetics, as I'll show you. So what Tatsuya did is he wanted to know what was the important region of OOPS1 and SPE11. So he made CRISPR GFP fusions, and then he proceeded to make in-frame deletions throughout both proteins. And he coded them based on whether they were viable or fertile. So for SPE11, for OOPS1, this region is required. In fact, he could make what he calls GFP OOPS1 mini, And this is necessary and is sufficient for function, this small little protein, with the caveat it's made at a higher level. He did the same thing for Sp11, and this region was important. Remarkably, in Katie's model, if we look at the metrics of the statistical metrics of the AlphaFold predictions, the ordered regions map to the required regions. This was really surprising to us and suggests that maybe there's some truth to this, and we're going to try to use these models to make specific point mutants to test this idea further, in addition to doing structural biology. Now, one of the things that Tatsuya noticed was that there was this deletion D. When he made these deletions, I asked him, can you put them at 25 degrees, please, and see if any of are temperature sensitive? And remarkably, this little tiny 14 amino acid deletion is temperature sensitive. So at 15 degrees, the worms are happy and they grow. At 25 degrees, they have a brood size of between 1 and 2. And so I did a suppressor screen and I isolated 40 rare dominant suppressors. Many of them seem to suppress very well. And here the dominance is not due to haploinsufficiency. I had to look at a lot. These are rare missense mutations that probably restore a contact with the defective region. And I should say that one of the reasons I wanted to do this deletion D was outside the region that we think mediates the interaction. So we thought this might mediate interaction with other proteins. So I isolated 14 rare suppressors, and thus far I've characterized 12 of them, and they define 3 genes. One was an allele of GSP3, which, as you remember, was one of the proteins that came down with both Sp11 and UPS1. And it's amino acids conserved in human PP1. It's in an extracellular loop. It predicted extracellular loop. And so we think that maybe this phosphatase promotes the activity of the complex, and some of the phosphorylations that we're looking at may be regulatory in the process. I also got 9 alleles of a gene which was a target of spin-4 degradation called chitin synthase, which is involved in making the eggshell, and 2 alleles of a gene called EG3. Now, chitin synthase and EG3 localized to a cortical complex called the egg complex. And this contains genes that are required for fertilization and eggshell formation. Chitin synthase itself, mutants of chitin synthase affect polar body formation. And this was a conundrum for the field because these are channels. Chitin synthase is a channel, and it makes the chitin layer between the zygotic membrane and the vitellin membrane. So it was hard for the field to really understand how what's happening out here could affect polar body formation out here. So what we're thinking is that there may be a cortical recruitment model. Maybe after fertilization, UPS1 and Sp11 find each other. Then they dock at the cortex by interacting with the egg complex. And then they're in the right place for cortical actin dynamics for completing meiosis. And in turn, they may activate the components of the egg complex for fertilization, for eggshell formation. So what I just told you is that in the worm, there seems to be paternal control of oocyte meiotic maturation and early embryonic development in several ways. So in one way, MSP promotes oocyte meiotic maturation as a signal. UPS1 and Sp11 seem to be required for meiotic cytokinesis. So Sp11 has to find UPS1 and be required for meiotic cytokinesis. And then the MEMI proteins discovered by Martin Strako seem to be required for meiosis too, and they also interact with GSP3 and 4, which are involved in probably the UPS1 Sp11 complex function. So there's paternal control here, but at least for the case of UPS1 and Sp11, there is a conundrum here. The conundrum is in 1996, Susan Strom and Heidi Browning did this interesting experiment. They had cloned Sp11 and they put the cDNA under control of the heat shock promoter. They injected adults after spermatogenesis happened, and they could transiently heat shock the animals and get like 10 viable progeny. This suggested that Sp11 can be provided by the oocyte. Okay, so we wanted to revisit this issue because why has evolution taken, if Sp11 is provided by the sperm and OOPS1 by the oocyte, why has evolution separated them into different gametes? Seemed to be a question. And in this experiment, we couldn't analyze the efficiency. How efficient is this process? And also you can't see where the protein is. We wanted to revisit this with the modern tools that are available at the time. So what Tatsuya did is he used the MEX5 promoter to express Sp11 in the oogenic germline, and he also used sperm-specific promoters to express OOPS1 in the sperm germline. And when we do this experiment, actually what you can see is that OOPS1 and Sp11 now co-localize. So they normally co-localize after fertilization, but when you make them ectopically express them in the germline, now they'll interact, and this is dependent on OOPS1. And so we were able to do crosses to see the efficiency of this function because we could feminize the germline, and if we feminize the germline with this mutation and mate them with the Sp11 mutant, of course nobody hatches. But if we express Sp11 in the female germline, that's the only place it can come from, and mate it with the Sp11 mutant, now we get like 96% viability. So it's a very efficient rescue. By contrast, OOPS1 seems to be required in the female germline. We cannot restore function. So it remains to be understood, why has evolution done this? And it seems to be a paradox that we're kind of interested in. And we're also interested in figuring out what this complex is doing for its vital roles. So this is the team that did this work. So at University of Minnesota, we have Carolyn Spike, Tatsuya Tsukamoto, Micah Gerhardt, Erica, the very talented undergraduate, Katie Wallstrom, a retired professor who is relocated to Minnesota from Florida and is working in the lab. And we've collaborated with Erin Osborne Nishimura on the maternal mRNA clearance, and then Amy Jaramillo-Lambert on the work on OOPS1 Sp11. And we're hoping to continue these collaborations for years to come. With that, I'm going to stop and take questions. Yes. Okay, that's a very good question. So she's asking whether we can, the individual proteins can interact. So we'd like to do those experiments. So the difficulty is making the proteins in pure amount and we're learning to work with intrinsically disordered proteins. And as I tell the students, unfortunately, and the people in the lab, it's trial and error. And one of the things, these proteins are a little bit difficult to work with because when they're at high levels, they seem to phase separate, and so they don't bind to affinity matrices. And so very well. So we're still doing those studies now. Katie is a crackerjack biochemist, and so having her in the lab, that she's working on this with us. So she's also enjoying her retirement and just jetted off to Italy. She'll be back in March, so we won't have much progress till then. But we're trying to do those studies. And then the other rate limiting step is the actin studies. There's a dirty little secret about studying actin. Sort of most of us, like, we use stuff we buy from NEB and stuff, and you go in the freezer, you can't freeze actin. Well, you can freeze actin, but it just won't function after you freeze it. So you have to, so they actually go to Trader Joe's and get flash frozen chicken, and they isolate chicken. It has to be flash frozen from Trader Joe's, isolate the actin, and then all the experiments have to be done before the actin dies. You freeze it, dead. So yeah, so we want to make these proteins in pure amounts before we go back and make that. Yes. Okay. Okay. Oh, there are no phenotypes that we now appreciate, but we went down a deep, dark rabbit hole. So the deep, dark rabbit hole is that when we got those strains, we sent them to our collaborators, and our collaborators said, those strains are sick. And I said, no, the strains are not sick. So I asked Tatsuya to get them out of the freezer and passage them. They had a transgenerational effect. We were going, aha, the Sp11 localizes to the RNA halo of the sperm. Maybe it's bringing in RNAs, okay? So then we said, okay, let's test it. We expressed its Sp11 with 3 other promoters in the oocyte, and there were no transgenerational effect. And then I said to Tatsuya, I think it's another mutation. Can you separate it, please? And he separated it, and it went away. And so it was like rabbit hole. So we don't know. So some of the speculation is maybe OOPS1 has a redundant role in the distal arm of the gonad where it's really expressed, and that maybe Sp11 interferes with this, but it's something that we can't comprehend right now. We can just speculate. Maybe further study will illuminate this, but now we know that it's not just 10 animals, it's very efficient. So it's a conundrum, but there must be a reason. Yes. Oh, we haven't, I mean, they're viable. I mean, so when you arrest the oocytes, I think Tim Shadle and I had this idea that some of the first oocytes there aren't so good. And it's really hard to do that experiment because you have to get the ones out that were first. So we can't really answer your question. We don't know whether it required fertilization. So I will say he did purify. I glossed over this. He also did purifications from Sp9 animals, which are defective in fertilization, but are not defective in ovulation. And he got the same RNAs, and so we just combined the data sets. And it was really hard to do because SPIN4 is expressed in the distal germline, and it's expressed in that first few oocytes. So there's not a bunch of RNA. So he had to amplify the RNA before RNA sequencing. Unlike the OMA1 and Lin41, where you can just see, you get a GAB and you can sequence it. So when in that Venn diagram I showed, he went back and redid those experiments using the same protocol. So we can compare apples to apples. But yeah, that's a really good question. We don't know. Yeah. Great question. So the dynamics of the process. So one sperm enters and it gets diluted 1 to 10,000 fold. So when Susan Strom did her experiments with Sp11 antibodies, she can't detect Sp11 in the embryo. When we look at M. scarlet Sp11, we can't detect Sp11 in the embryo. So what we're doing now is we're halo tagging Sp11, and I have a colleague named Lexi von Diesman, who does single molecule dynamics, and we're hoping we can see, we might get a good signal to noise because we could fertilize an oocyte that has no signal, and we can label the sperm by treating the males with the halo tag fluorescent compound, and then maybe we could do molecular dynamics. That's what we're hoping to do, and so we need to get the halo tag, and so we don't have it. I did some injections, I didn't get it, but Tatsuya did the same number of injections, and it looks like he's gotten it. So I shouldn't do experiments anymore. Yeah, so the thing is meiosis is happening on the other side. So this point of sperm entry determines the posterior that was shown by Bob Goldstein many years ago, but at the anterior is always where meiosis happens. So even if you fertilize them at the wrong end, that will be posterior, but that's where meiosis will always happen. So we think it's maybe affecting dynamics globally, but what's happening in, Frank McNally proposes that cortical forces are actually driving the force that pushes the spindle through that. And we're trying to do, it turns out making these movies in utero with what he shows is much easier than using actin probes. We're trying life act and things like that, but it's not as easy as it sounds on paper. So we're trying to do those studies.